Good afternoon, and thank you for joining us. My name is Leonora Johnson, and I am the Director of the Office of Science Policy, Engagement, and Education and Communications here at NHLBI. We welcome you today to launch and move forward our Sickle Cell Awareness Month. This is the webinar series. It's titled Sickle Cell Science, Path to Progress. In this series, we aim to address some of the educational and information needs shared by us with the sickle cell community. Today's webinar is entitled Genetic Therapies in Sickle Cell Disease. We hope to give you an overview of genetic therapies, what they are, how they work, and how genetic therapies are being explored to prevent or treat diseases. Today's webinar is also highlighting activities of the Cure Sickle Cell Initiative, an NHLBI-led collaborative research effort aimed at accelerating the development of genetic therapies to cure sickle cell disease, and features a patient perspective on participating in clinical trials and the importance of safe and widely available cures. Today we have joining us as presenters Dr. Mark Walters, Professor of Pediatrics at the Jordan Family Director, BMT Program, Interim Director of Research at UCSF Benikoff Children's Hospital, which is located in Oakland. We also have Dr. Tracy Mondoro of the Translational Blood Science Research Branch within our Division of Blood Diseases and Resources here at NHLBI. And then our final perspective will be Lindrick Holmes. Lindrick is a patient participant and a representative. With that, I'd like to first remind all of the presenters to please mute yourself if you're not talking. For those that are participating as listeners, if you have questions at any time throughout the session, please use the chat feature on the WebEx and send your questions directly to the host. There's a drop-down that enables you to send your question to everyone or someone. In this case, we'd ask you to send it to the host. You may also forward questions to email address here at NHLBI, and that address is NHLBInews, one word, at nhlbi.nih.gov. We'll take some time to answer your questions once we've gotten through all of the presentations. A final note with regard to process and technology, if you have any experience that's challenging or technological difficulties today, I would encourage you to try to log back in using Chrome or Firefox. Those browsers seem to work better on this platform. As a reminder, today's webinar is the second of 4 webinars that we will host on Wednesdays from 1 to 2 p.m. during the month. You can find more information about other sessions by searching Sickle Cell Science Path to Progress on either Eventbrite or the NHLBI website. Again, thank you for joining us. At this time, I'm going to invite Dr. Mark Walters to begin his presentation. Dr. Walters?
Yes, can you hear me?
Are you there? We can hear you.
You can hear me? Thank you.
Yes, thanks.
Well, thanks very much for the kind invitation to participate in this this webinar series, and in particular to Dr. Tracy Mondoro at NHLBI, who's leading the Cure Sickle Cell Initiative for the NIH. Next slide. So I wanted to highlight some of the approaches to curative therapies for sickle cell disease today. Allogeneic hematopoietic cell transplantation using bone marrow or the blood that's left over after the birth of a child, umbilical cord blood, or a newer method where we coax the stem cells out of the bone marrow where they normally reside into the bloodstream, called mobilized blood stem cells. All 3 have been utilized in sickle cell disease and in some ways are the gold standard of the curative therapy for this disorder. The exciting future has to do, though, more with gene addition therapy, where an anti-sickling beta globin or the fetal globin gene is introduced into that hematopoietic stem cell harvested from a person's own blood stem cell to elicit a curative effect. This is experimental therapy, but looks very promising. There are also gene editing, where you change a gene in one stem cell directly to increase the fetal hemoglobin for the beneficial effects this might cause. And then there are gene editing approaches to correct the sickle mutation itself, which obviously should be a cure if that's converted from sickle to healthy. What I'm not going to talk about today are in vivo gene editing techniques to correct the sickle mutation, which are in some ways the most exciting, but not yet under clinical development. Next slide, please. So I thought I would begin, even though next week I see you're gonna have a session about bone marrow transplantation, just a quick schematic about how a bone marrow transplant is performed. The panel on the left is a cutout from a cartoon of a blood vessel, and in each blood vessel in our circulation are red blood cells that carry oxygen to the body, white blood cells that help fight infections, and then a smaller type of white blood cell called platelets that control bleeding, stop bleeding after you have a cut. So the way a bone marrow transplant works is that the mother or seed cells, the hematopoietic stem cells that give rise to those red blood cells, white blood cells, and platelets in the circulation actually grow and divide in the bone marrow, which is the hollow liquid part of the blood system in the middle of a bone like the one that's shown on the slide. Those are extracted in liquid form and then transferred to a transfusion bag where they're administered to the transplant recipient in the same way as a blood transfusion. So those bone marrow stem cells know where to circulate in the body, land in the bones of the recipient, and begin to grow and replace to make healthy blood cells, healthy red blood cells, and correct the sickle cell anemia. Now, in order to do this successfully, one has to administer very high-dose chemotherapy, both to destroy the sickle cell producing cells in the body before the transplant, and also to paralyze the immune system to accept the cells from the healthy donor. And ideally, the healthy donor will have the same transplant type. That is, will be immunologically, that is, the immune systems will be compatible, identical, or as close as they can be. Otherwise, there can be complications. And one of the important complications that's highlighted in that blue figure on the right are skin rashes and infection, which are complications of a condition called graft versus host disease, where the immune system of the donor attacks the recipient, and is the leading cause of transplant-related problems, including dying of the transplant itself. So it's a complicated, intensive therapy, but it's the same principle that will be applied to gene therapy, in which case, instead of using a healthy donor's bone marrow, a patient's own bone marrow will be used as the source of the donor cells. Next slide. So why isn't bone marrow transplant the universal cure for sickle cell disease? Well, as it turns out, only 18% of families will have that fully matched, ideally matched bone marrow donor. And when we look for an unrelated donor who by chance has inherited the same transplant type as our sickle cell patient, only another 19% will have a well-matched unrelated donor. So even if we wanted to treat every patient by a bone marrow transplant, only about a third will have a donor. In addition, because of this problem that I've just described, of graft versus host disease, and this small risk of dying of the transplant itself, some clinicians are reluctant, and patients as well, to proceed to a transplant. And so for this reason, until lately, transplant is largely restricted to children and applied very sparingly, so not really at all a universal cure. Next slide. So what are the approaches that might elicit a universal cure? So there are two approaches, primarily gene addition, where we add a gene to that hematopoietic stem cell, that mother or seed cell that gives rise to all the blood cells in the circulation, including the red blood cells, or gene editing, where we make a change in an existing gene in the hematopoietic stem cell to elicit a curative effect. So in the gene addition category, it's possible to add a healthy beta globin gene to equal the beta S, the sickle globin level, or inhibit the effects of the sickle globin, to add a fetal globin because it's anti-sickling, it interferes with the formation of those long strands of hemoglobin, the sickle hemoglobin that form in the red cell after the oxygen is delivered to the tissues, which deform the red cell, make it stiff, damage it so it doesn't last as long in the circulation, and cause all the hallmarks of sickle cell disease, which are anemia and pain. Express a fetal-like beta-globin gene because it will have anti-sickling activity, adding that, or reawaken the fetal globin gene with gene therapy. There's a way to add an anti-gene, if you will, an anti-effect to activate, reawaken the fetal globin gene. The gene editing approach, changing an existing gene in the hematopoietic stem cell, targets a gene that allows reawakening of the fetal globin gene, or using CRISPR or other techniques to correct the sickle gene correctly. Next slide. So I think you've already heard about fetal hemoglobin or hemoglobin F and how it's when its level is increased in a person with sickle cell disease, it helps prevent sickling, making the sickle cell disease milder. It's one of the targets in how hydroxyurea has a benefit when it's given to a sickle cell disease patient, and raising it has become a therapeutic target. Raising the level of fetal hemoglobin, awakening it after it's been silenced, has been the basis of several new investigational studies for sickle cell disease. So how does this work exactly? Next slide. So this is a picture, a cartoon of the type of globins that are made before birth and after birth. So follow the orange curve, which has the Greek symbol gamma. So that's the fetal hemoglobin, and that's the only type of beta-like globin gene that pairs with the alpha to make hemoglobin before a baby is born. So the gamma globin gene is perfectly healthy in a person, in a fetus with sickle cell disease, and so there are no problems before the baby's born. This is why we have to rely on newborn screening to make the diagnosis of sickle cell disease at birth, because the examination of the blood and of the baby would show no signs of sickle cell disease whatsoever. But look what happens right after birth. The green beta globin gene shoots up, it's turned on, and this is the one that carries the sickle cell gene, and the gamma globin gene is turned off. So certainly by 6 months of age, there's very little of that gamma globin gene available. So the gamma globin gene, which elicits an anti-sickling effect, that effect is lost, and that's when the symptoms and signs of sickle cell disease emerge in the first year of life. Now, by chance, some individuals have an inherited condition whereby the gamma globin gene isn't turned off at birth. It persists after birth. And in those individuals, if they inherit the sickle gene with that, they have a much milder disease. So we've learned from nature that these natural causes that raise the fetal hemoglobin will have a beneficial effect. Next slide. So how does this work? And this is a pretty wonkish slide, so bear with me. I'm gonna walk you through it. So there are 3 types of oxygen-carrying molecules depicted. The purple is the regular adult hemoglobin. That dashed yellow line is the fetal globin, and then the blue is an oxygen-carrying protein called myoglobin in the muscle, where the oxygen is needed for energy and for development. So in the lungs, in that pink shaded area, all 3 types of oxygen-carrying proteins are fully saturated, 100%. They're fully loaded with oxygen. But as the blood circulates and reaches the tissues, there's this natural decline in that purple loading down to 50% in the tissues, so that the oxygen can be, if you will, transported up to the yellow and blue curves where the oxygen is needed for growth and development. So before a baby is born, since they can't breathe the air, they don't have lungs, all of the oxygen transfers from that purple adult hemoglobin to the yellow dash fetal hemoglobin, in the baby circulation, and then from there to the muscles in the fetus so that the organs and other tissues can grow and utilize energy. So you can imagine that even if that fetal hemoglobin persists, there's still room throughout the travels of the red blood cells from the lungs to the peripheral tissues to deliver oxygen to that blue line, the higher one, for growth and development. So that's why persistent or elevated fetal hemoglobin isn't damaging, and in fact can be quite helpful in counteracting the negative effects of the sickle hemoglobin. Next slide. So how would this work in a gene therapy? So it begins at the very top with a virus depicted in red that has all of the genes the virus needs to cause an infection, destroy the cell it attacks, and then reemerge from that cell that's been destroyed to infect another cell. So beginning with that harmful virus, a disabled virus is created in green. So that's no longer harmful because all of the pieces in the middle of that strand, that squiggly strand in the middle, those represent genes that have been removed that are necessary for infection and causing harm. And all that's left is that dark green dash at the very end that will enable that virus's DNA, the genetic material, to be inserted into the DNA of the target stem cell. So into that disabled virus, the rescue gene is packaged. So it could be a fetal globin gene, it could be a healthy beta globin gene, and then that's incubated with the stem cells from the patient. So the stem cells are collected, those are the purple cells, and then co-incubated, thrown in a tissue flask together so that the packaged, altered, disabled virus can now insert that DNA into the stem cell. The stem cell takes up the DNA. It doesn't land in the sickle gene, but it lands someplace in the DNA, which is the genetic material of that stem cell. And then those stem cells that have been altered, now with a rescue gene inserted, those cells are injected into the recipient so that they can begin to grow and have this positive curative effect. Next slide. So how well does this work? This is an ongoing clinical trial sponsored by an industry partner, Bluebird Bio Incorporated, with several different transplant and sickle cell centers in the US and Europe. And this shows the results to date. These are patients who have undergone the procedure and then have their hemoglobin type monitored in the blood from 3 months through 15 months after the infusion of those modified stem cells, after the gene addition therapy. So this is an anti-sickling hemoglobin, and it's depicted in red. The green is the sickle hemoglobin, and the blue is transfusion hemoglobin. And if you follow the path of that pink line, the rectangle with the pink, it increases from 33% all the way to 59% in that one patient who's been followed now 15 months post-infusion. And what it looks like to us is that this is more like sickle cell trait than sickle cell disease. The green is effectively diluted, and in fact, since this hemoglobin has anti-sickling activity, it's even better than that. So these patients who have this experience, as far as we can tell, no longer have any signs or symptoms of sickle cell disease. They don't have anemia. They're not having painful events. And while it's too early to call this a cure, if it were extended for 5 or 10 or 15 years, I think everyone would agree that this would be a cure. So this is a very interesting and exciting result. And again, it could be universally available because we're using as the donor a person's own stem cells. So the sickle cell patient is the donor for the gene therapy modified stem cells. So can't get graft versus host disease. There won't be any problems with immune reactions in either direction. This is just regrowing cells that now carry a healthy copy of that globin gene to outcompete the sickle gene. Next slide. Now, this is really wonkish, so bear with me. We did a clinical trial some time ago where we attempted to do a bone marrow transplant in a safer way as an outpatient, just using a little bit of chemotherapy with the idea that we could get a mixture of both the donor and the recipient cells in the bloodstream. This has been perfected more recently by a team at the NIH, but when we first tried it, we ran into problems, and it's shown by that graph on the left. So by several months after the transplant, we could detect only 2 to 5% donor cells in the bloodstream. But despite that, if you look at that curve with the open round circles, that's the level of the sickle hemoglobin, and it settled out at around 50%. So during that period of time when we were limping along with just 2 to 5% donor cells growing in the bone marrow, because those healthy red cells that were derived from the donor stem cells had a long lifespan, they outcompeted the sickle red blood cells and effectively, for a period of time, eliminated all of the symptoms of sickle cell disease. Eventually, those few stem cells are rejected, but the principle was that we didn't have to correct all the stem cells in the bone marrow. We only have to correct, as it turns out, about 20% of them. And that's shown by the panel on the right, where the blue, which is the percent sickle hemoglobin, you can see it's right at 50% when they're in red are 20% donor cells. So 20% donor cells, if you can correct 20% of the stem cells and eliminate the sickle gene in those cells, you should have a very strong effect, if not cure. So that's the principle underlying gene editing, because it's going to be very difficult to correct all of the sickle genes in all of the stem cells. But if we can correct 20% of the genes in 20% of the, at least one of the genes in 20% of the stem cells, then that should be enough. Next slide. So this is the approach with CRISPR. I know that many of you have heard of this. The schematic shows an aqua-colored elongated protein that's anchored in place by a yellow sequence called the PAM sequence that adds to the specificity of this reaction. And then the proofreading activity, the way this is so specific for one particular DNA sequence in the target cell is through an RNA molecule, what we call a guide RNA. In this case, once everything's lined up, the aqua protein makes a cut, a double-stranded break in the DNA, and if you supply the donor DNA, which after the sickle allele is cut out, and the same machinery will add the right sequence and correct the mutation. So that's the last piece of DNA at the very bottom with the green healthy sequence inserted. So this has been successfully accomplished in all kinds of organisms. It's quite a fast reaction. It's very efficient, simple, and potentially inexpensive. Next slide. So this is my last wonkish slide, but again, I think I can walk you through the principle. So there's a team of us, supported actually by the Cure Sickle Cell Initiative, who've attacked this problem. So it's a disease team, it's a project, and this is an experiment done by my colleagues, Wendy Magus and David Martin, at the Research Institute in Oakland at UCSF Benioff Children's Hospital. So what they did was make the correction in stem cells, blood stem cells that we had donated to us from a person with sickle cell disease, and ask the question, after you made the correction, injected those stem cells into a mouse that accepts human cells, and then after 20 weeks, long enough to get blood or at least bone marrow cells from those human cells that derive from a true stem cell, so you have to wait 5 months or so to be able to say that, and ask what type of blood cell was enriched after the correction. So the panel on the right are some stem cells that went through the attempting to insert the healthy copy of the DNA did insert part of it, but not enough of it to correct the sickle allele. So these still have sickle cell disease. And what's shown is that luckily that only happens about 2 or 3% of the time. And the bone marrow on the left is not really different from the red cells. Those are depicted by the label CD235A. So no changes there. But look what happens where the sickle allele has been corrected. So now this is healthy globin gene, and so it's much higher. We're getting 20 to 25% of the marrow or stem cells corrected, exactly what we need to target for a curative effect. And look at the red cells, the CD235A positive cells. 50% of those have the corrected gene. So just as we predicted, based on our experience in bone marrow transplant, there's this natural enrichment of the healthy corrected red blood cells in the circulation. And it looks a lot like sickle cell trait to me. So we're quite excited about this result, and we're going through all of the steps needed to ensure safety and potency, but we think we'll be in a position to begin to enroll patients in a clinical trial in the next year or two. So that's where that project is. Next slide. And how would it work? Well, we would collect stem cells with that instrument at the top left by a process called apheresis. We would then remove the red cells and the platelets from that stem cell collection and then enrich the stem cells. That instrument on the right does that. So we put all the blood stem cells in a culture bag and then apply an electrical current. That's the day 2 package with the gene editing components. So the CRISPR-Cas9 and the rescue target DNA. And then after putting them back into culture for a day or 2, infuse those into a patient who's received a high-dose chemotherapy drug to make room for the gene-edited stem cells to grow. So this is how the clinical trial would be conducted. Next slide. Now, gene therapy is also being developed for other disorders. This is a product called Zolgensma for a uniformly fatal disorder in childhood called spinal muscle atrophy. But look at the price tag, $2.125 million per dose. So that would be quite challenging if that were the price of gene therapy in sickle cell disease, because it's much less rare than spinal muscle atrophy. And it's a concern that all of us should have. Next slide. So in conclusion, I think the gene therapy is going to work. I think the early results are very exciting, but I worry that the availability and affordability of the treatment will be a limiting factor. So I urge all of the health policy interested folks, that the sickle cell disease advocates, family members, work with us right now. Let's get working on this right away to ensure that once we have that universal cure in hand, that we can begin to give it to the persons who we think would benefit from it. Thanks very much.
So thank you, Dr. Walters, for that great talk. My name is Tracy Mondoro, and I'm the Chief of the Translational Blood Science Branch in the Blood Division here at NHLBI. And I'm here to talk about the Cure Sickle Cell Initiative. So NHLBI has funded, next slide, please. NHLBI has funded much of the breakthrough research that has led to improvements in treating sickle cell, as well as the transplant biology that led to one curative strategy. The Cure Sickle Cell Initiative has been established to build upon all of these successes. And our efforts are guided by our desire to build upon, as I said, decades of research, but we want to further explore novel genetic therapies as a potential option for people living with sickle cell disease. And we know that genetic therapies may not be for everyone, and that focusing on cures and using the word cure may seem overly ambitious, but it's our hope that we can come together as a community to accelerate the development of safe and effective new approaches that will allow people to live free of sickle cell disease. Next slide, please. The Cure Sickle Cell Initiative is working to bridge the gap between the research and those that are impacted by sickle cell disease. So NHLBI is involving patients at every level of this initiative. In essence, our goal is to bring individual components of the sickle cell disease community together, including patients, families, providers, and advocates to help design and to participate in genetic-based studies to advance cures. Next slide, please. These are the folks who are leading the initiative. We have Dr. Gary Gibbons, the head of the NHLBI. We have Dr. Ed Benz, who is the President Emeritus of Dana-Farber, Dr. Leslie Silberstein at Boston Children's, Dr. Keith Hoots, who's the Director of the Blood Division at NHLBI, and there you see my picture at the bottom, the Chief of the Translational Branch. Next slide, please. We have, obviously we're not doing this alone. In addition to our other partners in the federal government and all of the investigators and patients, we do have some formal relationships with the California Institute of Regenerative Medicine, or CIRM, and the American Society of Hematology, or ASH, to collaborate in areas of common interest, such as co-funding projects and working with patients, providers, and laboratory scientists. Both of these organizations have dedicated substantial portions of their budget to work in sickle cell disease. We are pleased to report that CIRM and NHLBI are co-funding work that Dr. Walters is performing and just spoke about. Next slide, please. The initiative was launched in September 2018, and just a note about the other work that NHLBI does, the initiative will not replace any ongoing efforts of NHLBI. Instead, it will complement the Institute's broader sickle cell disease research investments in basic clinical, translational, and implementation science. NHLBI will continue to fund research being conducted by investigators focused on sickle, and this initiative will help fill the gaps that cannot be covered by traditional funding methods. So after some stakeholder meetings, the following goals were established. Next slide, please. So I'll get back to the goals in a minute. I just wanted to note that we're not looking for one curative strategy. As Dr. Walters stated, transplant is a cure when a donor is available, but there are multiple approaches through gene therapy and gene editing that Dr. Walters alluded to, and the Cure Sickle Cell Initiative wants to bring as many of these forward as possible and not pick a winner, because we think that different patients will require different strategies. And ideally what we would like to do is assemble a large body of data on different approaches or variations of some of the approaches, because we want to be able to give these therapies to adults, but we also want the FDA to allow us, once they're very safe, to scale down to children, because if children could be cured before when they're young, then some of this irreversible organ damage, such as kidney or vision or cardiovascular, would not occur, and then the cure would be complete with no side effects as we see in some adults. Next slide, please. So we are currently exploring, as I mentioned, different things that can't be funded through regular NIH grants. And so we want to focus on defining safety and risks of these different therapies. We want to look at potential off-target effects. In other words, how can we make sure that the sickle cell gene is corrected and no other genes are harmed? We want to identify any hurdles in the technology that's used to give these therapies. And we want to look at best practices and methods to do things such as develop laboratory tests or assays so we can confirm that the therapies work, create standard operating procedures to isolate and modify cells so every product is made exactly the same way, just like the aspirin you buy at the drugstore. We want to produce common data elements, and this is so data from different trials can be compared to each other to decide which therapeutic strategy would be best for a specific patient. And we want to design patient-centric clinical trials with meaningful endpoints. So some of the endpoints in trials that you may have read about are laboratory measurements that can indicate the sickle cell, that your cells are no longer sickling. But we want endpoints that are evident to a patient. For example, if shortness of breath were reduced after you received a therapy, then that's something that would positively affect a patient's life. And as Dr. Walters just mentioned, we want to assess the clinical and economic impact of genetic therapies on patients. So what are we funding? We're funding animal studies to show safety and dosing limits with different kinds of therapies. We're going to be funding manufacturing studies, again, ways to make the best and most consistent products in large quantities. We want to find safer conditioning regimens, so the preparation for transplant or gene therapy can have adverse side effects, so we want to find ways to reduce the toxicity of these procedures. And diagnostic tools, such as an MRI scale, so parents could be assured that there have been no further changes in their child's brain or sort of quantify a stroke risk after a curative therapy. This would apply to transplant or gene therapy. And we're also supplementing ongoing trials to make sure they have adequate funding to complete planned enrollment and to be able to follow up with patients to record how they're doing after receiving these therapies. Next slide. This is a graphic showing some of the things that we're doing in the initiative. We are reaching out to patients. I mentioned common data elements, manufacturing. Also, the way we're funding this initiative allows us to work with the companies and to help them where they might be stuck, either in things they need for the FDA or assisting them with economic data, which we mentioned, to figure out how to compare the cost of living with sickle cell to the cost of curing sickle cell, and also working with patient and advocacy groups. Next slide, please. So we do want to engage the patient and the advocacy community. And to do this, we are engaging patients to participate at all levels of the initiative. We have patient representatives on our executive committee, on our steering committee, on our subcommittees. We've had some listening sessions, and we also want to form some advisory groups for the initiative, including patient scientists, including young adults. We think that's a group that we really need to hear from, because these are some of the folks who may just be coming of an age where they can qualify for these trials, to give us advice on how to reach out to other patients. And then a survey was published, and the citation is at the bottom, but there have been surveys and other methods to reach out to patients to find out how they feel about genetic therapies. And investigators from the genome research published a survey on patient attitudes and beliefs towards genome editing. And so the responses confirmed that the patient community is skeptical regarding new curative strategies. And after seeing some of Dr. Walter's graphs, one can certainly see why. I mean, this is new, gene editing and gene therapy, it sounds a little bit like science fiction. Plus, we know that there have been past disparities in access to medical care, in clinical trials, and in treatment of people with sickle cell disease. And so the conclusion of this paper was that the advent of genome editing has renewed hope for the sickle cell community, but caution tempers this optimism. And so it's the intent of the Cure Sickle Cell Initiative to, moving forward, to maintain and invigorate this optimism, but we want to demonstrate that we're listening and acting on the promising science, as well as remembering what we're hearing from the patients and keeping the patients continually engaged as the science moves forward. Next slide, please. And now I'm pleased to introduce Lindrick Holmes, a patient representative who has kindly agreed to speak at this webinar.
Hey, how everybody doing? Thank you for having me. I'd like to start off by talking about what options we have other than the curative therapies that are available now. First thing I like to state is I understand that the sickle cell community, the patients are not a monolith. Not all stories are the same. So I'm gonna try to, talk about my perspective alone. But I see a lot of similarities that we all suffer from. As far as the treatment options that were available to me, there were hydroxyurea, which I had some side effects taken. I was on it since like 5th grade. There were blood transfusions, which, can give you iron overload and stuff like that. And then as an adult, they kind of like just drop you, and you have to figure out everything on your own. And and that's a trial in its own, having to give reason why you should be treated. That's how it feels. It feels like you have to give a reason why you should be treated, why are you struggling with this, and it's very hard to deal with. When I first heard about gene therapy, I was a little bit skeptical too, but I had a realization that over time, if I wait and try to see how many people go up and get them to perfect this and, you know, get their stuff together before I give it a try, by that time, I need a hip replacement or a stamp for my aneurysm in my brain, or, you know, I lose some organs. I'll be on dialysis, and I wouldn't have no quality of life at all. So for me, it was do or die. And, you know, I think one of the main things that I hear a lot of people talk about, one of the things that they're skeptical about is like the vector that they use to try to correct our gene, a part of the trial that I've been a part of. And I mean, it's not really too much to be afraid of, because they take apart that virus and they just use it as a vehicle to try to get the corrected gene to burrow itself and implant itself into your bone marrow, so you can, you know, not have sickle cell. Since I've been, I know that curing is a term that I hear some professionals, you know, a little leery to use, but I feel cured. And honestly, I didn't even understand the level of how bad sickle cell actually was until I got on this side and I actually had the ability to be cured. You know, it being fatigued all the time, being in pain all the time. I didn't realize how often that was until I got a chance to be on the outside looking in, like I have now. I feel like it is like very important that we have participants come be a part of the trial so that we can move forward and have this widely available. There's no way that this can be widely available if we don't have people partaking in what's going on here. If everyone's apprehensive about it, then, I mean, what other options do we have? The closest thing that I believe that I found through the trial to be a treatment is blood exchanges. Blood exchanges, I never even heard of them until I got up on this trial, because they don't have anything like that available here where I'm from in Mobile, Alabama. So I think that's the closest thing that we could get to some sort of stability. Hydroxyurea works for some. It didn't work for me. Blood transfusions are dangerous, but blood exchanges is where they take out a percentage of your blood and then they put back in some healthy blood cells, and for a time or two you feel kind of normal. But it's an entire different feeling when you actually cure. Like, I actually have way more energy that I've ever had in my life, even when I was going through the trial and I was getting blood exchanges. So, but to my knowledge, I think that is like what is best. But without participants, without people becoming brave enough, and I hope not desperate enough to join or be a part of this, we'll never get to the point to where we're actually free of sickle cell. And I don't know, I'm just tired. I'm just tired of having to, go back and forth with. I was tired of having to go back and forth with, doctors and hospitals, and in and out of pain, starting all over again, getting an apartment, getting a car, and then having a sickle cell crisis, and having to start all over again, and having organ damage and having AVN, and a lot of that stuff is just like, you know, it sticks with you regardless if you have the cure or not, you know. So I feel that it's in the best interest of the community for us to push this forward, to just push this forward and to just keep hope alive. And this is the only hope that I had. I didn't have anything like a clinic where I can just go to get fluids or blood here in Mobile. They don't have clinics like that for adults. They do for kids. But once you get to a certain age, they don't have that available for you. So I was kind of left out here for the wolves. But that's pretty much just my testimony. I'm pro gene therapy, and I feel like everyone needs to, you know, at least have an ear out for it, because we don't really have much else besides this.
Lendrick, this is Leonora. Thank you so much for your comments and your testimony. We appreciate it, and I think it speaks volumes to what we do. At this time, I also like to thank Dr. Mondoro, Dr. Walters as well, and we're now able to take some questions from our listeners. So again, you may use the chat function on the WebEx platform, or you can send your questions directly to NHLBI News, one word, at nhlbi.nih.gov. So we do have a couple of questions. The first one is for Dr. Walters. Can you briefly explain the difference between gamma and beta and why gamma turns off?
Sure. So the gamma, that's just a Greek letter, is the nomenclature for fetal hemoglobin. So the fetal hemoglobin is on for birth because the baby doesn't have lungs that breathe air. It gets all its oxygen from the mom's circulation. So it needs to bind that oxygen tighter than the mom's hemoglobin. So that's why it's on before birth. And then after birth, when the lungs start working, the fetal hemoglobin isn't made anymore, and then there's this shift to making the adult type beta globin. So that's, I hope, answers the question.
Thank you. And then another question for you had to do with, do you know how much it costs, how much gene therapy approaches cost?
Yeah, the price of these therapies hasn't been established yet because they're not yet approved by the FDA. They haven't been licensed. But we do get a snapshot of what it might look like, but we just don't know. So there's another hemoglobin disorder called thalassemia, and gene therapy is also pretty far along towards approval for that. And the price tag we've seen in Europe, it's not approved yet in the U.S., but it's nearing approval in Europe. The price tag we've seen is close to $1 million. So it's daunting, and these are going to be expensive. The price is based on what it costs to care for a person with the underlying disorder, lifelong, and to save a life, you know, if there's a risk of dying early of the disease. So those are always difficult price tags to work out, and then how to get insurance to be able to cover the high cost of care in a single bundle. That's also being attacked in a way that there may be ways to spread out the cost of the treatment over a longer period of time to make it a bit more affordable for the insurers. So it's kind of a long answer, but to a short answer would be I just don't really know the cost of what it's going to be.
Okay. Thank you, Dr. Walters.
Yeah.
A question for Dr. Mondoro. How can people with sickle cell disease find out about clinical trials that they might be eligible for?
Well, that's a very timely question. So you may have heard there is a website called clinicaltrials.gov, and we understand, I've been on that site. I find that site confusing, and I've been on it, you know, probably 50 times in the last year. So it is a good resource, but it's hard to navigate. So the initiative is working on trying to find out how, for instance, people in Oakland, because there's good publicity around Dr. Walters' trials, so we're trying to find out how people in cities where there's more than one trial, how they're finding out about it, how they're interacting with the trial physicians, and then we want to make that available on our website probably for everyone to see. So right now, talk to your doctor or your medical provider or your friends or your support group. And right now, clinicaltrials.gov, unfortunately, is the best that we have, but we are looking for ways to improve that.
Thank you. Lindrick, a question for you. Lindrick Holmes, how long was the process for you in the trial participation?
It took about 2 years. It took about 2 years total. There was a lot of changes that happened along the way. They were still learning as they was going, so anytime they thought that there was something they could have did better, they'll reschedule and they'll come back and do it better. So, yeah, it took about 2 years for me to go through it.
And then another question was, do you experience any pain following the clinical trial in which you participated, so post-therapy pain?
The only pain I guess you feel is like, and it's not really like nothing to be afraid of kind of pain, it's the engrafting pain of like the stem cells engrafting into your bone marrow, to your bones. So, but that, to me, that was a good pain. That wasn't a bad pain. I was like, yeah, it's working. I knew it was working once I started feeling that. And everything after that, no pain, no sickle cell related pain whatsoever. I stubbed my toe on something, I feel it, but that's it, no sickle cell.
Thank you. I also want to follow up on Dr. Monduro's answer about participating in clinical trials. The NHLBI has a Center for Health Information. We call it the NHLBI Qi. They will also answer any questions that you might have, as well as help you navigate and find appropriate trials near you. They also are able to refer you to our Office of Patient Recruitment within the NIH for assistance in finding whether or not there are applicable trials here at the NIH. And that number is 301-592-8573, or you can send an email directly to NHLBI-info, I-N-F-O, one word, at NHLBI.nih.gov. A next question, Dr. Walters, has there been any progress on the use of gene therapy in Africa?
Yeah, that's an area where we need to be more proactive. I'm not aware of any gene therapy trials active in Africa, and that's because there are some significant challenges to exporting it and getting it active there. But we need to put more energy into finding out how to accomplish that.
And then either you or Dr. Monduro, what do you think doctors might need to know about upcoming treatments, and when might they be available?
Mark, I'll let you handle that.
Oh, thanks so much. Well, it's I hope the audience gets the sense that within the constraints of a webinar, how excited we are about these developments. I mean, there's significant movement, in part because of really good support from both the NIH and organizations like CIRM, California Institute for Regenerative Medicine, and other foundations to accelerate these curative therapies. So the best way to stay abreast of them is to go to the national meetings, the hematology and the sickle cell disease meetings. And in addition, it's possible for individual sickle cell centers to request one of us to make a visit and give a talk and update about the results. But honestly, none of these therapies is quite ready for launch in the community and making them widely available. That's going to take a longer period of time. So, it's going to be a concerted effort, I think, both from the regulatory agencies and the industry sponsors, and from the hospitals and doctors who are going to deliver the therapy, to coordinate the running out these therapies as they become available and getting the patients treated.
And I can add to that in our conversations with the Food and Drug Administration, they understand that we want to move this forward as quickly as possible while keeping rigor on safety studies, but they are very willing to work with investigators to figure out how to check off all the safety boxes and not let things, linger, and get them approved as quickly as possible, and to tell us what needs to be done so that we can do it. So they're very excited as well to work with us.
And then I did want to follow up a question on gene editing and gene transfer. Dr. Walters, are there any side effects to gene editing or gene addition?
Yes, there are several potential side effects. So this is a population of blood stem cells. So this is the source, the lifelong source of all the blood one has in circulation. And so you can imagine if you're altering the DNA code in the blood stem cell, if there was what we call an off-target change, that could have dire consequences. So, for example, what if you damaged a gene that you need for that stem cell to continue to divide and give rise to other blood cells? And if you lost that stem cell, so that's a problem. Or what if you caused a DNA change, either from gene addition or gene editing, that awakened cancer-causing gene? And what if that took a long time to have that effect because it was a rare event? Well, we're just not going to have information about that level of safety for a period of time. So there are some theoretical concerns about the long-term, safety of these kinds of treatments. So we build into our plans as best as we can rigorous tests of minimizing the off-targets, doing a really careful job of screening and monitoring for the off-targets after we've delivered the therapy, and so forth. And the good news is that so far we haven't seen a problem, but that's something we're always worried about. Then there's the side effects of the preparation before you infuse the gene therapy or gene-edited, the modified stem cells. And right now we're relying on very high-dose chemotherapy to wipe out, destroy the sickle-producing cells in the bone marrow. Well, that has side effects too. You can imagine that, that's the source of your immune system, so for a period of time, a person would have limited protection against infection, so you have to accommodate that. It causes thinning of the lining of the mouth, that causes mouth sores, again, temporary, but still a problem. And then we have observed so far at least one case where the chemotherapy in and of itself caused a leukemia, which is a blood cancer. So we know that the very high-dose chemotherapy will cause infertility in adults who are exposed to high-dose chemotherapy. So lots of side effects, actually, that I just have to gloss over in a short talk, but something that we go through very carefully and extensively whenever I meet with a family or a patient before we make a decision about whether or not to pursue the treatment.
Okay, thank you for that, Dr. Walters. There was a point of clarification that just came in, and the question is, if I understand correctly, hemoglobin 206 treated patients do not experience pain, and what age group are these patients?
Yeah, that's the Bluebird Bio-sponsored trial, and a small cohort of patients have been evaluated for pain, but that's right. The ones in this last cohort of patients using all the optimized conditions and preparation don't appear to be having painful episodes after the infusion.
Okay, thank you. I do want to let people know that we will position information or a link to clinicaltrials.gov on the page that where you'll also find the other information about the upcoming webinars. And that just to let the community know that the NHLBI and working closely with the Cure Sickle Cell Initiative will be continuously developing educational and information that's pertinent to this area. And so stay tuned. As that information comes out, we'll definitely take the opportunity to share it with you. I would like to, it is 2 o'clock, and I would like to end on one final question, and that's to Lindrick. Post-treatment, what's the thing that you least expected, and what's the thing that you are most happy about?
The thing that I'm most happy about, I don't think it's appropriate for me to answer, but
fair enough.
I think I just glossed over it by saying me and my wife are happy, and that's things that I'm really, really happy about. The thing, I didn't really have much to expect post-treatment. I mean, they had everything that they warned me about it. Probably could have happened, but to be honest with you, I was just grateful that one of those things wouldn't be sickle cell. Whatever it was, right. It wouldn't be sickle cell. So I had no sickle cell problems, and I guess it's a surprise to everybody else. I'm relatively normal, aside from, like I said, the organ damage. Like I got avian, and I still have an aneurysm and stuff, but that's it. Nothing. Just pretty normal guy.
Well, thank you for that. I'd also, for those that want to see more, hear more about Lindrick's profile, it will be posted on NHLBI.gov, and if you go to the search and search Faces of Sickle Cell Disease, you'll find a Lindrick story there to share. And thank you for sharing that, Lindrick. With that, I'd like to bring to an end our webinar session today. Again, thanks to all for joining us. We hope that you enjoyed learning about how NHLBI is exploring genetic therapies to prevent sickle cell disease. Again, if you still have questions or any lingering questions, please feel free to email nhlbi.news@nhlbi.nih.gov. Also, don't forget to join us next Wednesday for the next webinar in the series, which will discuss bone marrow transplants, other therapies, and sickle cell disease. Join the conversation between webinars on Twitter using hashtag #ScienceOfSCD. Thanks again for joining us. Good afternoon.